now. I will admit
Yes,
all the students.
all students. Okay, thank you, Bari.
Yeah.
Thank you, Bu Alami.
Yes, you're welcome, Bu Jongkon.
So today the moderator will be Bu Alami.
Pardon?
The moderator will be Bu
Oh,
Alami,
okay,
not
okay.
me. That's
Okay.
the problem.
I'll help Pak Arif to be a moderator today.
Okay, thank you.
Okay. Okay, how about the participants?
Let me see.
Are all of the participants coming to the Zoom? Maybe we just wait, yeah. Bu Jongkon.
Yes. All participants camera is off. Maybe we need to ask them to turn on the camera.
Oh, sorry, Arif. I forgot to close the door. So, okay, I'm back.
Yeah, no problem.
So you are in home now, Bu Jongkon.
At my office, Bu Alami,
Oh, okay.
because I have the problem with my laptop, and then I have to come to my office first to use my PC in the office.
Okay. Okay. Is it far from your house, your office?
It's around 10 minutes.
10
for
minutes.
driving, around 10 minutes.
By drive a car. So you
Yeah.
go to the office by car. Okay.
Yeah.
It's quite
How-
near from
So
the
reach out.
No, it's all and my car is too old. How many students do we have for this class?
About 90.
90.
Yes, 90 students.
90 students, and they are in the same year.
Yes, but still a half of all of participants, still 50.
Don't worry, we can wait.
Yes, Bu Jongkon?
It's also for my student. Sometimes we need to wait around 5 to 10 minutes because
Okay.
I know because some students maybe they have the problem about the internet connection.
Internet connection. Yes, yes, that's true. For the students, would you turn on the cameras, please? So Bu Jongkon and Bu Jongkon can see your face, yeah. All of the students here.
Yeah, now I can see many beautiful and handsome students.
Only the students,
Of course, also the professor. Oh, they are so young.
Yes, because mostly they are in the third semester. Four or third semester. Yeah, third semester.
The third semester, it means they are in the second year of bachelor.
Yes, second year. Yes. 70 already. Maybe Pak Arif can chat to the group to remind the students again.
Yes, thank you. I will.
How about the guest lecture program in your university, Jongkon? Is that any like this? I mean, the guest lecture from other university.
We also have the guest lecture from other university, but depend on the course and depend on the budget. We how can I say it first depend on the cost and depend on the budget that we ask for. I mean, actually we need to ask for the budget before the it's like a plan. And for example, for next year we need to ask for since this year for the budget for the guest lecture or something like that. And maybe next semester I try to search for the way to invite Pak Arif for to be the guest lecturer here. But I need
Thank
to wait.
you.
I need to wait until the semester that my course is open or something like that. And
Yes,
for the
but you don't need to think about the salary or the money.
we have to try first. I know because it's not much money, but sometimes it's like when you do something for us, we also need to give you back. But also for me, I don't care about the money, Arif. And when you ask me, I'm willing to join your course and also to meet your very nice students. Since last year, it was my first time to meet your student, and I really appreciate it because the student is, how can I say, they're pay attention to the course and ask the questions. And some questions I also don't know the answer, but it also let me like I need to study more about the knowledge in microbiology. So I also thank you all of you to invite me.
Thanks a lot, Jongkon, because yeah, you're so so helpful. Yeah, we have done doing this guest lecture series. It's about three. We have 3 meetings here, and Jongkon willing to help us, and then sharing a lot of knowledge for us. And yeah, hopefully maybe if you have a plan or something like, yeah, maybe invite Pak Arif to your university. Maybe it will strengthen our both of the universities like this.
I think so, and I also think about the research collaboration, not also the lecture.
Yes. Thanks a lot.
You're welcome. You don't need to say thank you. I'm willing.
Okay. How about if we start for the guest lecture today, Pak Arif?
Yes.
Yeah. It's about
I think we can start. It's almost 80. Yeah.
Okay. Okay. I will start this guest lecture series for today. So this is actually our last session of the guest lecture series that delivered by Dr. Jongkon Saising from the School of Health Science, Mae Fah Luang University of Thailand. So Dr. Jongkon Saising, welcome again in our course in this our virtual guest lecture series today. And today I become a moderator as a representative of the lecturer that responsible in these microbiology courses. So the aim or the purpose of this guest lecture series is it can maybe the main purpose is we can build the collaboration between both of the institution, Mae Fah Luang University Thailand, and then the biology department of ITS. And then the second, of course, we can broaden our knowledge and then our insights, especially for the microbiology theme. And then for today, Bu Jongkon will give will explain about the last topics. This is natural products as antimicrobial compounds. And for Bu Jongkon, we have about an hour for Bu Jongkon to explain or to share about the PPT or the slide regarding the natural product as antimicrobial compound. And then after that, it is continue with the discussions sessions. So without any further ado, let's we give a warm welcome to the Dr. Jongkon Saising. So time is yours, Bu Jongkon.
Okay. Thank you very much, Bu Alami and Pa Arif, for inviting me to be a guest lecturer here. And today is the third lecture, and which is the last lecture. I will talk about the natural product as antimicrobial agents. From last week, we also learned about antibiotics and mechanism of action of antibiotics, and also the antibiotic resistance, which is the major problem worldwide. And one of the method that or one of the alternative approach to help for antibiotic resistance is to find another or alternative way, and the natural products is one of the major sources for antimicrobial agents or to search for the new antibiotics. And as you can see here, for natural product, there are many sources. For example, from plants, in this figure, as you can see here, and in the plants, there are many compounds and many metabolites that exhibit antimicrobial activity, and also the natural products in the group of the animals. And everyone know the egg and also the honey. In the egg and honey, there are the substances or the compounds that show to inhibit or kill the bacteria. And also in the group of the mushrooms, as you know, some mushroom have the substances. Some substances are toxic, but some substances can be used as the antimicrobial agents. And from algae, we also can find the compounds that can inhibit or kill the bacteria. And of course, from the microorganism, as you can see from last week, our first antibiotics called penicillin is from the microorganisms in the group of fungi from penicillium. And here, this paper, they conclude or they reveal about the natural products as antimicrobial agents, and they are from the group of the plant. And I just show you, for example, the plant for potato, the potato peels. There are the compound, the major component. For example, the component called chlorogenic or caffeic organic acid that have the bacteriostatic effect. I think for this word, you know from last week, the bacteriostatic effect, it means the compound can only inhibit the bacteria but not kill. And this compound have the effect on the bacteria or E. coli or Salmonella typhimurium. And for example, the tomato, I think tomato, the vegetable that everyone know, and in tomato, the major component, the seeds of tomato, the major component, for example, the metabolites such as the fatty acids or carotenoid or saponin or the compound in the group of phenolic compound. And these components has the activity, antibacterial activity against the bacteria, such as in the group of Staphylococcus aureus and Epidermidis, and Micrococcus luteus, the Enterococcus faecalis, and also the yeast named Candida albicans. And here you also can study more yourself, follow this reference that I all of my my handout or my document I will send to Arif, and he can distribute to all of you, and then you can study more yourself. And this table is the antimicrobials or the agents from animal origin. And for example, the milk from the cow, and in the milk there is the antimicrobial agent called lactoferrin. And the lactoferrin has the activity, antimicrobial activity against the important pathogenic bacteria such as E. coli, especially O157H7, which is the dangerous E. coli for can cause the disease in human, and also Listeria monocytogenes, the Salmonella, they like the foodborne pathogen and can cause the disease in human. And some substance that I think everyone have heard before, the chitosan in the animal in the group of exoskeleton of the crustaceans, in the for example the shrimp or in the group of crustacean, and the chitosan also inhibit the bacteria. For example, E. coli or S. aureus. One of the example that I will show you is lysozyme. The lysozyme that I'm not sure you have the experiment in the lab in microbiology, and this lysozyme is in hen eggs. It is in hen eggs and also mammalian milk. And actually, the isosan are also in our saliva, our tears. But in the case of animal origin, we can find the lysozyme from hen eggs and mammalian milk. And in hen's eggs is especially in the white part of the hen eggs. We can the lysozyme can be found, and it can inhibit or kill the bacteria, especially in the group of gram-positive bacteria. And these are the antimicrobial agents from animal origin. And apart from plant, from animal, the bacteria itself also can be the source of antimicrobials. For example, the agent called niacin. The niacin can be isolated from the bacteria called Lactococcus lactis, and this compound has the activity antimicrobial activity against Clostridium and Listeria monocytogenes. And as you can see here, there are many agents. For example, lactocin, bulgaricin, or pentaricin. And you can notice here, most of the antimicrobials from bacterial origin in this table is from the bacteria in the group of lactobacillus, which is lactic acid bacteria, and they can produce many types of antibacterial agent or antimicrobial agent. And this is the reference of this table, of these three tables. And for today, I will focus in this kind of plant because it's one of the plants that I study during my PhD and also my postdoc when I work together with Arif. But some of my result here is from during my PhD experiment. And for this kind of plant, it's called, the name is, in scientific name is Rhodomycus tomentosa. I don't know the name in Indonesia, but in English name, the normal name is like rose myrtle or downy myrtle or something like that. And this is the picture of the plant, the flower, the leaf, and also the fruit. Actually, from the report, this kind of plant also can be used in Indonesia. There's the report that they use this kind of plant, the decoction of the leaf and the root of this plant is used for diarrhea and stomachache treatment. And in Malaysia, there is also the report that the leaf, the leaf of this plant, the crushed leaf, can use to dress the wound. And actually, from my review, this plant, I will show you the next slide first. For my study, I use the leaves of the plant and dry the leaves. And also after the leaves were dry, I extract with the ethanol. After the extraction, we will get the crude extract. And from the crude extract, we use many techniques. Actually, not me, but one of my colleagues in chemistry department. He isolates the compound from the leaf of this plant, and this compound called rhodomytone. And actually from the report, this rhodomytone is not first isolated by our group, but this first report of the isolation of rhodomytone from this plant is in the group in Indonesia. Is in the group in Indonesia. And then I study the activity of the crude extract and also the compound, the main compound from the leaf of this plant called rhodomytone. Normally, when we get the compound, when we get the extract, we need to screen for the antimicrobial activity. I mean, for me that I'm interested in the microbiology, so I screen for the antibacterial activity of this plant extract. It is the screening method. We use the method called this diffusion method to screen whether the crude extract have activity against the bacteria or not. The compound, not the compound, the plant extract, I mean the ethanol extract, is dissolved. The solvent that I use to dissolve the compound is the MSO or dimethyl sulfoxide. And then after the extract dissolved well in dimethyl sulfoxide, I drop. I'm not sure you can see this figure well or not. I drop this extract onto the disc. As you can see here, we have the blank disc. It's like the paper disc, the circle paper disc, and we drop this compound into the paper disc, and we can get the disc that full of the extract. And after this disc will dry, when we wait for the disc dry, the another part of this method, I need to prepare the bacteria that we would like to test. In my case, and for this, this is the Staphylococcus aureus. I grow the bacteria on this kind of media, the triptych soy agar or TSA, for 24 hours. And then when we get the single colony, here, we pick the colony and grow them in the media called MHB or Muller-Hinton agar, and wait for 3 to 5 hours, and then we adjust the turbidity of the cell growth culture in MHB. We adjust the turbidity equal to the McFarland standard number 0.5, and with this turbidity of the culture, we use the sterile cotton swab and spread the culture here into the media called Muller-Hinton agar, and then we place the disc here that we already prepared. We place the disc on the surface of the Muller-Hinton agar that we already spread the culture of the bacteria. And after that, we incubate this plate at 37 degrees Celsius for 16 to 18 hours. And after here at 16 to 18 hours, you can see on the plate. You can see on the plate the bacteria grow. The bacteria can grow, and you can see the colony of the bacteria. And you also can notice the zone surround the zone surrounding the disc here. You can see the zone without the growth of the bacteria. This zone is called clear zone. This zone is called clear zone or inhibition zone. It means the bacteria surrounding the disc cannot grow, and you can see the clear zone. From this clear zone here, we measure the size of the clear zone. Measure the diameter. You measure, for example, from this size of the clear zone, and measure across the disc. And now you can get the size of the clear zone. If you can see the clear zone, it means this extract can inhibit the growth of the bacteria. And the result of this experiment are shown in this table. This table is the screening activity of the ethanol extract of the plant Rhodomycus tomentosa, and I test against the Staphylococcus bacteria. And this bacteria I isolated from acne lesions, and I divided the Staphylococcus into two groups. In the groups of coagulase positive and the group of coagulase negative Staphylococci. I think if you learned before about the identification of Staphylococcus in the group of Staphylococcus, and you can divide the Staphylococcus into two groups according to the coagulase test. And I test in the group of coagulase positive 64 isolates and 85 isolates for coagulase negative Staphylococci. I measure the inhibition zone or the clear zone of every tested isolate. And as you can see here, there are many sides of the clear zone. And the percent of the number of the isolates that when I measure the clear zone surrounding the disc, if less than 10 millimeter in the group of coagulase positive Staphylococci, it is around 14%, and 82% of coagulase positive Staphylococci. I can measure the clear zone between 11 to 15. And the most of also coagulase negative Staphylococci, the size of the clear zone is between 11 to 15. This is the results from this diffusion method that we use to screen whether the crude extract have anti-bacterial activity or not. And also another example of the results of this test, I also test this crude extract on the bacteria called Propionibacterium acnes that isolated from acne lesions. And for this bacteria, I can isolate 21 isolates and 18 isolates the inhibition zone of the extract against these bacteria. The zone is between 15 to 20 millimeter. And this is the example of the test that we use to test the antimicrobial activity of the crude extract of natural product. And then when we know this crude extract has the activity against the bacteria that we would like to study, then the next experiment, we would like to know the concentration of the compound or the extract that can inhibit or kill the bacteria. So we need to determine the inhibitory minimum inhibitory concentration, we call MIC, and the minimal bactericidal concentration we call MBC. The MIC is the concentration of the compound or of the extract that can inhibit, just only inhibit the growth of the bacteria. But for MBC, is the concentration of the compound or the extract that can kill, cidal mean kill, the concentration that can kill the bacteria. The method that we use called microdilution method. It is broad microdilution method. And we use the broad microdilution method. We use the plate called 96 well plate. Actually, this kind of method can be done in the also the test tube, but you need so many test tube. If you do this method in the test tube, we call macro dilution method. But for micro dilution method, we do it in the 96 well plate. We can do many compounds, we can dilute many concentration. It is easier and more convenient than macro dilution method, and use the test tubes. For the first step, we need to prepare the extract. We need to prepare the extract, and we dilute the extract. For example, in this experiment, I start in the first column, here, with the concentration of 1024 microgram per mil. And then I make 2,4 dilution. Of the compound. So in the second column, here, the concentration of the compound is 512, and then we make this 2,4 dilution until we come to the last well in the plate. And then you add here in each well, I add 20 microliter of the compound in each well. We will have 20 microliter of the compound at different concentration that we already diluted. And then we add the medium, the culture medium called Muller-Hinton broth into each well. We add 80 microliter of MHB into each well. And then we add the inoculum. The method to prepare inoculum is same as when you prepare here. You adjust the turbidity of the inoculum into McFarland standard number to 0.5, and then at the inoculum 0.5 McFarland standard, which is around 10 to the 8 CFU per ml. You dilute it into 10 to the 6 CFU per ml. And then you add this inoculum 100 microliter into the wells, and you mix them together. You mix the extract, you mix the culture media, and you mix the inoculum, and then you incubate this plate at 35 to 37 degrees Celsius for 16. I'm sorry for this. This is 16 for 16 to 18 hours. After incubation, you notice the growth of the bacteria in each well. The concentration, the minimal concentration of the extract that can inhibit the bacteria. I mean, you cannot observe the bacterial growth in the wells. That minimal concentration we can call MIC. And then in every well that you cannot observe the growth of the bacteria, I mean the medium in the well still clear, they are not turbid. In every well, you streak onto the agar plate, for example the TSA plate, every well, and at the concentration that the bacteria cannot grow on the agar plate, the minimal concentration that the bacteria cannot grow on the agar plate, we record as the MBC, or minimal bactericidal concentration. And after we finish this experiment, we can see the result, and this is the result from my experiment after I did the broad micro dilution method, and I can record my MIC and MBC values. I did in the ethanol extract, the pure compound rhodomytone, and I also add the antibiotic, the effective antibiotic called vancomycin, which is the effective antibiotics used for Staphylococcus as a positive control. And I test in the bacteria Staphylococcus aureus and Staphylococcus epidermidis, 5 isolates and 1 the standard strain, and also 5 isolates and the standard strain. From the result, you can see the MIC of the ethanol extract against the Staphylococcus is between 32 microgram per ml to 256 microgram per ml. And in the case of rhodomytone, the MIC value is between 0.25 to 1 microgram per mil, and which is similar to the MIC value. For vancomycin, you can see the MIC values is between 0.5 to 2 microgram per mil. The MIC value of the extract or the compound and antibiotics also depend on the isolates or the strain that you test against the bacteria. And from this result, as you can see here, you can notice that the concentration of rhodomytone is very close to the concentration of antibiotic called vancomycin. It means this compound is very effective to inhibit or to kill the bacteria in the group of Staphylococcus. You can notice from the MIC value and also MBC value. One of another of the similar result, I test also this ethanol extract, rhodomytone, but in this table, I test against Propionibacterium acnes, and I change the antibiotic. In the case of Propionibacterium acnes, I use erythromycin because it is the antibiotics that used for Propionibacterium acnes for treatment of acne lesions. So that's why I changed from vancomycin to erythromycin for Propionibacterium acnes. And as you can see here, the MIC value, here you can notice 50 and 90, MIC 50 and MIC 90. MIC 50, it means the MIC value that can inhibit 50% of the isolate that we test. And MIC 90, it means the MIC value, I mean the concentration of the extract or the compound or the antibiotics that can inhibit 90% of the tested isolate. The MIC 50 of the ethanol extract is 16. Why the MIC 90 is 32 microgram per mil, and this is the range of the MIC value. It means when you test this compound to 21 isolates, the MIC values of this against this 21 isolate is between 8 to 32 microgram per mil. In the case of rhodomytone, as you can see here, the MIC 50 and 90 is 0.5. This is the range of MIC value. And for erythromycin, the MIC 50 and 90 is 0.03 and 0.12 respectively. So this two table is just to show you that the way to present the MIC and MBC value after you do the broad micro dilution method to determine the MIC and MBC value against the bacteria that you would like to test. the antibacterial activity. And one of the method that you can do to see the effect of the compound or the extract or even antibiotic to the bacteria, we call time QSC. The time QSC. Actually, it is very similar to the dilution method, to the broad dilution method here. But with this method, you see the result at the end point, I mean after 16 to 18 hours. But if you would like to know, I mean before the end point, before 16 to 18 hours, can the compound or the extract or the antibiotic kill or inhibit the bacteria before the end point? So we need to do time QSC or time Q study. In this step is the same step as when we do the microdilution method for MIC. For time QSC, we do it after we finish, after we can get the MIC value. After we get the MIC value, and when we prepare the extract, we prepare the concentration at MIC value, and maybe lower MIC value, only half of MIC value, and you also can increase the concentration of the extract into 2 times of MIC, or 4 times of MIC, or even 8 times of MIC value. And then you prepare the inoculum, same as in the method to find the MIC. But here, you need to increase more the volume of the extract, the volume of the culture medium, and also the volume of the inoculum. Only the volume, not concentration or not inoculum size, and then you mix them together. After that, you incubate at 37 degrees Celsius. And at time intervals, you collect the sample that you incubate at time intervals. For example, at 2, 4, 6, 8, for example, 11, 12, and 24 hours, depend on you. For example, at after 2 hours of incubation, you take the sample. And then you do the dilution of the sample that you take. After serial dilution, you put or you drop the culture dilution on the plate, for example, TSA plate, and you incubate again around 24 hours, and you can see the colony that can grow on the plate. You count the number of the colony. We call this step viable count. And then you calculate back from the dilution that you dilute, from the number of the colony in different time. You did it in different time. And from the number of the colony in each time interval, you plot the graph. And this is the example of the time Q curves. The time Q curves that I did for the ethanol extract. The upper one is the graph for the ethanol extract. And rhodomytone. I'm sorry, let me check here first. This is the graph for the rhodomytone. It is the rhodomytone against is aureus, the upper one, and is epidermidis in the standard strain. And you can see, for example, in the upper one here, this is the control. The control means I did not put the compound. It's only the bacteria and the culture medium. And you can see the bacteria can grow from at the 2 hours to 24 hours. The bacteria can grow. And when they add, in the case of the tubes, that they add the compound in different concentration. For example, in 2.5 MIC or 1 MIC, 2 MIC, and 4 MIC. With the compound, you can see the graph. Here, I'm sorry, here is the y-axis is the number of the bacteria represent in the log CFU per mil. And here the x-axis is the time that we collect, that we take the sample here to do the dilution. The normal, the control, the bacteria can grow like normal. But when you add the compound, and you can see the number of the viral cell is reduced. When you add more compound, the number of bacteria reduce faster. For example, when you add 4 MIC of the compound, the number of the bacteria reduce at 4 hours. But if you add less compound, you need more time to kill or to reduce the number of the bacteria. Same as in the case of Staphylococcus epidermidis, the control. And here, when you add the different concentration of the compound of the rhodomytone, and this is the way that you present the results after you do the method called time QSC or time Q method. And you can notice the effectiveness of the compound against the bacteria in time intervals. And this one also I did it in the Propionibacterium acne, and you can see, for example, in the control, and when you add the compound, the bacterial cells is reduced. The bacterial cells were reduced. And this is the method or the technique that you can study the antibacterial activity of the extract, the compound, or also the antibiotics. And for this kind of method, we test against the bacteria in the planktonic cells. And from the knowledge in microbiology, I think everyone know that actually the microbes behave two modes. They can behave like planktonic cells. The planktonic cells is mean they float or swim in some liquid media. And they can behave as biofilms. The biofilms which are attached to the surfaces. This is this figure will show you the status of biofilm development. As you can see here, this is the bacterial cell. They act as planktonic cells or planktonic bacteria. Then these bacteria can attach to the surfaces. The surfaces can be biotic surfaces such as the human tissue or human matrix cover the in-well device, or even this surface can also be abiotic. For example, the polystyrene or the glasses or something like that. And after attachment of the bacteria to the surfaces, this bacteria can produce many kinds of protein, and they accumulate and form like the matrix here. We call this is the biofilms, the biofilms of the bacteria. And after the biofilm, after the biofilms are formed, these biofilms can the planktonic bacteria can also disperse from the biofilm. When the bacteria can form the biofilm, I mean they can live together, they can accumulate together. There are many good points or many advantages of the biofilm forming bacteria. The biofilms can protect the bacteria from the antibiotics or from antimicrobial agents. They can protect the bacteria from the toxin and also the host immunity. Imagine that when they live together like this, the antibiotics or antimicrobial agents or toxin, it is difficult to destroy the bacteria inside the biofilm. And also from our host immune response, it is also difficult to destroy the bacteria in biofilm. So in my work, after I test the activity of the compound or the extract against the planktonic cell, I also test the activity of the compound against the biofilm, and this flow chart is the method just show you the method to test the activity of the extract on the biofilms of Staphylococcus. For this step, is similar, similar to prepare the inoculum of the bacteria of the Staphylococcus. And then after inoculum preparation, I add this inoculum size of the bacteria into the microtiter plate and let them grow at 37 degrees Celsius for 24 hours. And after that, I watch this plate with phosphate buffer saline or PBS. Here, the bacteria can form biofilm in the plate. I mean, in this step, after incubation, the bacteria can form biofilm in the plate. And I watch the unattached cells in the plate. I watch the planktonic cells. So now I have the biofilm that attached to the surface of the microtiter plate. And then I added the culture medium and the extract. Added culture medium and the extract. And I incubate for another 24 hours. 24 hours. And then I add the reagent called thiosolium blue tetrasodium bromide or MTT. Or MTT. This MTT will react with the cells. It means if the bacterial cells are still alive, the MTT will change the color, and then we can measure the absorbance of the color. I mean, after we add the MTT, after we add this reagent, we discard the reagent and we add the MSO to dissolve the crystal. After we get the reaction from the MTT, and then we measure the absorbance at around 600 nanometer. Actually, it is around 570. And at that time, I use 595 because of my equipment. I can use at this wavelength. And this is the result from this experiment. This is the effect of the ethanol extract. And as you can see in the white bar and the rhodomytone in the gray bar here, and the antibiotic vancomycin in the black bars. And as you can see, we also increase the concentration. We also increase the concentration of the extract, the rhodomytone and the antibiotic vancomycin. This graph show you the percent viability of the cells after treated with this extract, the compound and antibiotic. And you can see the viability of the cells reduce after you add the compound or antibiotics. The viability reduce when you increase more concentration of the compound, especially when you see the viability cells after treated with rhodomytone. In the case of this strain, you can see at around 16 times of the MIC, rhodomytone can reduce the viability of the cells more effective than the antibiotic vancomycin. You can notice here when you compare with vancomycin from 16 times of the MIC value. And also in the case of the standard strain of is Staphylococcus aureus. And this figure is for the bacteria Staphylococcus epidermidis in the clinical isolate and the standard strain. And you can notice here, especially in the case of standard strain, rhodomytone is more effective than vancomycin when you test with the biofilms of the bacteria. You can see the viability of the cells is reduced. And then we observe the biofilm. From these results, we also observe the biofilm using scanning electron microscope. And from these scanning electron micrographs, in the figure A is the control. I mean, no compounds, no rhodomytone or vancomycin. And the second figure, the second picture, figure B, after treatment with rhodomytone and after treatment with vancomycin. And for control, you can see the accumulation of the cells as the biofilms. You can see the thick layer of the cells as the biofilm. And after treated with 16 times MIC of rhodomytone, you can see the number of the cells were reduced and the layer of the cells is thinner when you compare to the control. And in the case of vancomycin, you can also notice the biofilm, but it is thinner than when you compare with the control. But in the case of rhodomytone, at 16 MIC, it is more effective to reduce the cells when you compare to vancomycin. So it means it's more effective than vancomycin in the case of to study in the biofilms of the bacteria. And here is the here are the example of my results when I study about the antimicrobial activity of the extract and the compounds which is isolated from plants. These are the examples of the study of the antimicrobial agents from plant origins. And from the review, right, in 2019, they review about the compound, many compounds in this kind of plant, in Rhodomycus tomentosa, and many kind of compounds that can be isolated from this plant, for example, from leaves, from stem, from bark and twig, from bud, also, and from flower and fruit. And you can see many compounds that can be isolated from this plant. This is the review. And also in this review, they also add the bioactive agents and also the biological activity of the agents in this plant. For example, there are many studies show about the result of ethanol extract from the leaves of the plant, and these are the results to inhibit or to kill the microorganisms, and also the study of the activity of rhodomytone against the bacteria, and also the mechanism of action. And I just show you here, and if you are interested in, you also can follow this review yourself, and all the examples. And as you can see here, the one of the alternative method or the source to search for the new or the alternative treatment to solve the antibiotic problems is the knowledge from the traditional healers. And from this knowledge, from the knowledge of traditional healers, we know that there are many kind of herbs or many medicinal plants, and we need to add the scientific validation to the many kind of herbs or medicinal plants, and with scientific validation, we can use these herbs and can develop to be the herbal medicinal products, and also the new antibiotics or new antibacterial agents in the future. And this is all of the example that I would like to show you today. Okay, if you have any questions or suggestion or comments, can. Okay, are there any questions from the students?
Okay, thank you so much, Bu Jongkon Saising. So this is the end of the slides, Bu Jongkon?
Yes.
Okay, that was a very interesting and clear explanation from Bu Jongkon, and I'm sure that we have already notes some of the things, some of the information here. That maybe the first from based on the explanation by Bu Jongkon, we can imagine that mostly or almost all of the food we eat is contain potential of anti-microbial compound, right? And then maybe for the species of the Rhodomycus tomentosa,
Yes.
it looks familiar, I think, in Indonesia, but I'm not sure about the local name in Indonesia, because yeah, maybe Pak Arif know, but I'm not sure about the local name, but it's look familiar, that species in Indonesia, but maybe I'm wrong, so I need to check it more about that.
I think you also have this kind of plants in Indonesia.
Okay.
The first isolation of this compound is from Indonesia.
From Indonesia, okay. Yeah, especially for the Thailand and then Indonesia itself is the well known as a mega biodiversity, right? So there are many of the natural product or like plant and then from the animal that have a potential to explore in its potential to become anti-microbial.
Yes.
And also Bu Jongkon have explained about how the method to measure and to determine the MIC and also MGC, right? So the students, I think, can learn about that. Okay, now we enter into the discussion session. I'm sure that the participants have already many questions to Bu Jongkon. You can raise your hands and give a questions. Ask the questions, please. Anyone? This is Z. Okay, please speak up, Z.
Okay, thank you for the opportunity. Am I audible, ma'am?
Yes, we can hear you, Al-Fawzi.
Okay, thank you, sir. Okay, I'm Alfonso Saxono from Microbiology Class A and have ID number 21. I want to ask about your publication about the potential of the rhodomitron. And fortunately, I found your publication in the Google Scholar, and I got the like your conclusion or something like that. And in your publication state that the ethanol extract and rhodomitron demonstrate a better ability than phenomycin to reduce the viability of biofilm growth cell. In here, what's the potential that we can utilize after we can conclude about this conclusion about the ethanol extract and rhodomitron for the viability of the biofilm growth cells? Thank you.
And so the question is?
I'm sorry. My question is what's the potential after we find out this conclusion like that?
Like the further usage or further utilization of the rhodomitron.
Yes, right, sir.
Okay. For this, I mean the further potential. I mean, from this result, we have our, other students also study the potential of this compound and also the ethanol extract to be, it's like from the result here, she continue to study, to formulate the product for acne treatment, because as you can see from the result, it has they have activities against Staphylococcus and also Propionibacterium acne. So one of the students, she developed the product for acne treatment, and some of the students they add this compound like into the for the coating of the gloves, and we can use the gloves like the antibacterial gloves or something like that. And some of the students they add this the extract into, how can I say, the food for the fish, because she studied more about the stimulation of the immune response in the fish, and that's why she add this compound in the food of the fish. And also other still in the step of the development of the product to use this kind of extract and compound.
Okay. How about the answer, Z? Is it clear enough, or do you have any confirmation about that?
Yeah, I think it's clear enough, and I want to answer part the question of maybe all of student here. What is we call this kind of place in Indonesia? We call as kamunting. So if maybe you don't know, and thank you for Mr. Ma'am Saising for answer my question. ขอบคุณครับ
ขอบคุณค่ะยินดีค่ะ welcome ค่ะปาซี
Okay, thank you, Z, for your nice questions. Okay, anyone else? You can raise your hand.
Maybe while waiting, may I ask a question?
Okay, Pak Arif.
I would like to know about the importance of the time-killing essay, because I think it's a kind of important essay, right? Do you maybe you can explain us why did you do the time-killing essay, not the other essay?
Okay, and if you see, okay, and from time killing essay here, you can see, I mean, sometimes you don't need to wait for 16 or 18 hours, and as you can see here, even sometimes when you add the compound and the number of the bacteria can be reduced when treated with the compound. For example, here in this figure, maybe if you add when you add the high concentration of the compound, sometimes with this method, you can, the first thing that you can see, you will know that at what time or at time interval the bacteria can be reduced. And the second thing is when you increase more concentration, you can see maybe the result of the numbers of the bacteria is not much different. And you will know that maybe you don't need to add more concentration. For example, when you add 1 MIC, 2 MIC, and 4 MIC, but when you can notice the result to reduce the number of the bacteria, sometimes you don't need to use higher concentration, because with higher concentration of the extract, it also higher toxicity to the human cells.
Okay, thank you. And can you correlate the time-killing assay with the growth stage of the bacteria? For example, the lag phase, the stationary phase, the exponential phase. Is it possible to correlate the result with this stage of the growth in bacteria?
Yeah, it is possible here. And let me yeah, you can see from, for example, in this graph, this is the control. This is the control, and it's like when you study the growth of the bacteria, and actually you can see the growth like the S-shaped curve. But here, here you see, this is like the in the log phase, the log phase of the bacteria, the lag phase here, the lag phase, the log phase is here, and this is the stationary phase. Here is the here is the stationary phase. And if you continue to grow this bacteria, I mean without adding the new media, this graph, the bacteria will be dropped here. It's like the S-shaped curve of the bacteria. And if the compound normally from the theory, the substance or the compound or the antibiotic will, how can I say, transport to go inside the cell in the log phase. And you can see when go to in the log phase and the number of the bacteria here declined in during this log phase of the bacteria also in this. So this is when we look the graph and compare to the phase of the bacteria.
Okay. So does it mean that the rhodomyrton is very active in the log phase when the bacteria is growing exponentially, right? Or can you say that?
I think you also can say that, but I think I'm not sure it's right or wrong, but in my opinion, or as far as I know, I mean, normally the compound or even the nutrient, the bacteria can absorb very well in the phase of the log phase. It is the normal phase that the bacteria would like to grow and they absorb the nutrient. And also when we add the compound or other substances or molecules, they will absorb well in this kind of phase, I mean in the log phase. But I think if you grow the bacteria until the stationary phase and you add the compound, I think the result may be different from when you add the compound at the beginning or at the log phase of the bacteria. That's why, I mean, from this theory, when we do the MIC assay or even this time assay, we need to grow the bacteria until around 3 to 5 hours for Staphylococcus aureus. But for the bacteria that they have, how can I say, more generation time, maybe the times to grow the bacteria is different, depend on the growth curve or the generation time of the bacteria.
Yeah. Okay. Thank you, for the answer.
Okay. Thank you, Pak Arif, Bu Jongkon. Yeah. So this is the interesting discussion today. Yeah. Maybe anyone else from the participants, if you have a questions. Bu Jongkon, in the previous week, Bu Jongkon have told about the characteristic of the antibiotic, which is consists of the bacteriostatic and then bactericidal.
Yes.
Regarding this, our topic today, that the natural product that can act as a anti-microbial is that characteristics also can be have by the natural product.
Oh,
the
yes. You mean the
bactericidal, yes.
Okay, thank you very much for this question. And then the student also can be observed or can be noticed from your question. Is really good question. Last week, as you mentioned, that we already learned about bacteriostatic and bactericidal activity, and you can notice this activity from the concentration of MIC and MBC. You can notice here is the concentration of the MBC and MIC is the same. You notice here is the same. This compound has bactericidal activity, or even the MIC is higher. You also can call bactericidal activity if the MIC value is not higher than 4 times. You got my point? For example, here is also higher. MIC value is higher than MIC value, but it's just two times higher. Also here, just 2 times higher. And in this case, you also can notice from vancomycin. Vancomycin is the antibiotic that exhibits bactericidal activity. You can notice here, the MIC value and MBC value is the same or equal, or even MBC higher, but it's not higher than 4 times. This is still 4 times. Yeah, and I will show you here. But for this table, it's not clear. But sometimes when you test, or maybe when I test the activity of the antibiotic, for example, erythromycin. When I test the erythromycin with Staphylococcus aureus, and when you get the MIC value, the MBC value of erythromycin is much higher than 4 times of the MIC value. So the activity of erythromycin is bacteriostatic activity.
Okay. Yes, it is clear now. Okay, thank you. Yeah. Hannah, how about to sterilize this this natural product? If maybe if we doing if we doing some of the, yeah, we check it and to know the potential, then how we can sterilize this product prior we doing the antimicrobial test. Maybe filtration or something.
Okay. No, normally I to be true in my case, oh, you mean when you would like to check the sterility of normally I just dissolve and do the experiment. But for example, here when you like for this case, you would like to check the sterility, right? I mean, you just the easy way for me is just use the sterile loop and take the sample from this extract and just streak onto the agar, for example, TSA. And if there is no colonies grow on this agar plate, I think it means this extract is sterile. I always do like this. And also when you do the MIC, I always have the wells that put the extract or the compound with the media. And if after incubation, when you observe the result, if your media is still clear, it means the extract is sterile.
Okay, so this is a simple one, yeah. To check about the contamination. Okay, thank you, Jongkon.
Oh, welcome, really welcome.
Yeah. Is there any question from the students or participants, or is it enough for the last session of this guest lecture today? Because the time is already almost 9 o'clock, yeah. Yeah. Okay, it's enough. Yeah, because Dr. Jongkon already give or share the clear explanation regarding this topic for today is natural product as antimicrobial. Yeah, so thank you so much, Bu Jongkon, Saising, for the really valuable information for our last session today. And maybe we have to close, yeah, this the last serial of the guest lecture today, and then hopefully maybe in the, or we can meet again in the next semester maybe, yeah, in the yeah, guest lecture or something, other program that like the collaboration between our both institution. And for all of the participants or students, thank you for your active participation during these lectures. And then thanks a lot. And I'm as a moderator today. Yeah, just want to end the serial of the guest lecture. Thank you so much. And
I also would like to thank you to you, Bu Alami, and also Pa Arif to invite me to be a guest lecturer here. And to be true, I'm really willing and I'm very happy every time when I meet both of you and also your students. And to be true, I'm looking forward to see all of you every time when I have the schedule here. And because your students is really active and concentrate to my lecture, and the question that they asked is very interesting questions. And I hope that I also will have a chance to welcome you, Bu Alami, and also Pa Arif to also be the guest lecturer in our university, I think soon.
thank you so much, Bu Jongkon, Saising. Yeah, see you.
Thank you very much. Okay, see you.
Thank
Bye-bye.
you all.
Bye-bye.
next
See you
time.
later.
Bye.
Okay.
Bye, every
Bye.
student.
Yeah, thank you.
Bye.
Maybe I will end the Zoom ya, Pak Arif.
Yeah, thank you.
Okay, thanks a lot. Thanks a lot, Jongkon and all.
Bye-bye, Ka.